Okay, we're switching gears a little bit here. This is a session on visualization, and I think the main point about visualization is that we wouldn't need it if we weren't trying to communicate stuff into our brains. So we're really talking about how do we get information and understanding into people. I'll thank the people that have been involved in some of the work that I'll be presenting, and I'm also going to present other people's work as well. I guess one question, if you want big questions, is should we think of ourselves like physicists think of themselves? Should we make bigger and bigger communities to solve larger and larger problems, or is biology a little different? And I'd like people to think about that somewhat, because you know, you can develop a monoculture in biology which is very fragile, but if you have a lot of ideas floating around, you get, I think, a richer environment to understand things. So I'm going to simply talk about very quickly three aspects and challenges that we face. One of them is simply the human interaction, and I'd like to make the point that we should use more than our eyes when we're talking about visualization. We understand things in lots of different ways, and we should start to think about how we can use those different ways to understand things, communicate things, and teach things. Secondly, the data interaction is important, and we've already heard about putting things together across scales. The fact is that evolution is scale-free. It happens at all levels simultaneously, and we need to think about what those implications are for our understanding. Now, I come from a kind of a molecular background, so I see things from the bottom up, and the thing that strikes me is that there is a continuity from physics to chemistry to biology, and there are no gaps physically. Everything works together. And I think that's the challenge that we have to think about. And again, from a physical-chemical point of view, we have to think about the fact that life does not exist at equilibrium. It exists in a highly non-equilibrium state. And so our simulations, especially even our biological molecular simulations, have to start thinking about simulations away from equilibrium. So for the human-computer interface, we know that biological structures are complex. We can't avoid that. What we can avoid, possibly, is the complications. We want to uncomplicate the complexity. And sometimes just the mouse and keyboard in our standard computer interface is not enough, and images may not be enough. So I quote Marvin Minsky, who's one of the main fathers of artificial intelligence, which is if you understand something in only one way, then you don't really understand it at all. So we need to start to understand things in a variety of different ways. This picture shows two crystallographers who use all the computational technology that they want, but they spend a lot of time working with physical models. And physical models promote lots of different cognitive, perceptual, analogical, and social interactions and thoughts and cognition. And so what we've been doing some in our lab is actually trying to go beyond the picture and start to deal with things in a physical way, because we're talking about really, truly physical phenomena. And so when we virtualize these things, I think that we lose some of the power of our understanding. So what I want to do right now is go to a little bit of a live demo. It's always risky, and whether this works or not is an open question. I guess you can see me here. Okay, so this is one example. This is a virus model. Some of you have seen this before. So this virus model is actually made up of viral assembly units. And as I shake it, because of the way I've analoged the interactions, you can see that it starts to assemble. And if I shake it long enough, it will assemble into its lowest free energy state, the shaking is analogous to temperature. As you can see, it's now come into two half shells. They may not fit together. They have to be dynamic. They have to break apart and reform. And with the 10 minutes that I have, I'm not going to try to assemble it wholly, but usually it takes between 30 seconds and 3 minutes. It's a stochastic process. Now, physical models alone are not sufficient, obviously. We use computers, and we're not going to stop using computers, but there's technology that exists now that allows us to integrate computational with the physical. So, for instance, here I have a little marker, and what I'm showing you, this is just some augmented reality. What I'm showing you is all of the amino acid side chains, the amino acid types in proteins. So I can take that and put it onto a physical model. And in fact, I can, because this physical model has the physical constraints, but I also have the constraints of the physical model. So this is actually a user interface in a sense. So the idea is that we should start thinking about different kinds of user interfaces that may in fact help us understand different phenomena. Just one little last example before I go back to the more standard slide presentation. Here's a model of a protein, and basically I can augment it with additional information like the electrostatic environment, and I can manipulate it vis-a-vis another protein and not only dock against a physical shape, but dock against the electrostatic complementarity. So let me just simply, oh, I've got to get to the right. Okay, so that's one aspect. Now, the aspect of integrating information, I'll briefly mention a project that Graham Johnson will go into in more detail later, which is called CellPack, which basically takes information, just like David Goodsell will describe in his talk, from a variety of different sources, but instead of creating a two-dimensional watercolor like you see on the left, it creates a three-dimensional packing diagram that puts shapes into a volume. And in fact, it can put different shapes into different volumes. It can populate surfaces, and it can make procedural models that make fibers and complicated cells, complicated structures in the same environment. Just briefly, this was a picture that David did many years ago, and it took me about a month to make that inset, which is a three-dimensional model. These models can now be made in seconds using the CellPack. And finally, you can, because it's automated, you can make micron-based, micron-size environments. I just want to mention the sources. I mean, we have to be concerned with what kinds of sources we have, and I think there's a lot of new technology that's coming along. Soft X-ray tomography is one that I think is important because no staining, a complete cell data set in 5 minutes, and high water contrast, so you can do segmentation very quickly. And this is just an example of the autofill or the CellPack, which looks at a synaptic vesicle and creates 3D models that essentially are segmented because you know what they are. You can use this if you can simulate the experimental technique as standards to actually measure how well your segmentation works. And finally, I want to make one last point about simulation. Most of the simulations at the atomic level are done in equilibrium conditions, like this Brownian dynamics. But in fact, these are dynamic systems, and I just wanted to mention as a last point that in fact, this is a system, an experimental system, that is made up of 4 different types of molecules. Basically, microtubules, kinesin, streptavidin to connect the kinesins together to multivalency, and PEG that makes fibers out of the individual things. And what you can see is actual behavior. So if you, on the left, there's no ATP, you add a little bit of ATP, and these things start moving. These are advection. And finally, more ATP, and you get, let me go back for a second. If you put this into a geometry like a bubble, then you start to get movement at different scales. At this scale, you get real movement. Small scales are frustrated, and you don't get any movement. In fact, if you put it on the surface of something, you actually get synchronized movement and behavior that from this very simple system. And I think that these kinds of systems are the kinds of systems that we should be using to test some of our simulations, our molecular simulations, because they're not at equilibrium. Currently, there's an energy flow through the system. So with that, I'll just briefly, again, acknowledge the co-workers in this, and thank you for your attention.